Your CQ values can look perfect, your amplification curves can look beautiful, and your conclusion can still be wrong. Here are 5 mistakes in qPCR gene expression analysis that catch almost everyone, and each one takes about 5 minutes to fix. Let's begin. The first mistake is assuming that a housekeeping gene is automatically a valid endogenous control. It isn't. A housekeeping gene and a reference gene are not the same thing. GAPDH, beta-actin, 18S. These can shift with treatment, hypoxia, cell cycle stage, even confluency. A true reference gene must be stable across your specific experimental conditions, and that stability needs to be demonstrated, not assumed. Normalized to an unstable reference, and that instability carries into every fold change you report. The second mistake is taking a reference gene straight from a previous publication without testing it in your own system. That study used GAPDH, so I will too. That's not a valid justification. Stability is tissue and condition specific. A gene that's rock solid in mouse liver can vary considerably in human neurons. Validate your candidate genes in your own samples, under your own conditions, using tools such as GNorm, NormFinder, BestKeeper, or RefFinder to rank their stability. Then normalize to 2 or 3 stable genes, rather than just 1. The third mistake is not fully understanding your statistics, starting with your error bars. An error bar shows how precise a measurement is, how far individual observations are dispersed around a mean. A narrower error bar indicates a more precise, tightly clustered measurement. A wider one indicates greater variability between observations. For instance, measurements in Group A are more precise than in Group B. But error bar isn't one single metric, and each type tells a different story. Standard deviation, or SD, shows the spread of your raw data. Standard error shows how close your sample mean is likely to be to the true population mean. It shrinks as sample size grows, so a large N can make your data look more precise than it really is. Confidence interval, or CI, is a range, typically standard error times 1.96, within which the true value is expected to fall with a given probability. In short, SD describes your data's spread, standard error, and CI describe your confidence in the mean. The same numbers can produce 3 very different-looking bars, so always state which one you're plotting, and use biological replicates as your true N, not technical ones. The fourth mistake is skipping controls and quality checks simply because the CQ values look fine. Genomic DNA contamination, degraded RNA, and primer dimers don't produce an obvious red flag. They simply shift your numbers quietly. Run a no template control and a no RT control, assess RNA integrity, and inspect your melt curves carefully. You should see a single clean peak, not 2. 2 peaks in your melt curve indicate primer dimers or unspecific amplification, for example from residual genomic DNA. This is the baseline set by the MIQE guidelines, and it's what separates a result from a reliable result. The fifth mistake is assuming 100% amplification efficiency by default. The 2 to the minus delta delta CT method only holds true if both your target and your reference gene amplify at a similar, near 100% efficiency. If they don't, your fold change values will be quietly inaccurate. Run a standard curve, calculate efficiency from its slope, and aim for a range of 90 to 110%. If your assays fall outside that range or don't match each other, use an efficiency-corrected method such as the FAFL model instead. A target amplifying at 99% alongside a reference at 78% is a discrepancy that only a standard curve will reveal. So, 5 checks before you trust a single fold change. Confirm your reference gene is stable under your specific conditions. Validate it in your own samples. Know whether your error bars represent SD, standard error, or CI. Verify your amplification efficiency, and run your controls. Do this, and your data will earn its conclusion. If this was helpful, subscribe, and let us know in the comments which mistake caught you. Thanks for watching.